Okay, today I'm going to talk to you about photosynthesis, and we'll remember that many organisms use light energy to make organic molecules, not just plants, but large multicellular alga and some single-cell protists along with prokaryotes, so like cyanobacteria and purple sulfur bacteria all use light energy to make organic molecules. If we're taking a look at the formula of photosynthesis, we'll notice it looks pretty familiar. It's really just cellular respiration turned around. Except in this situation, we're going to be using light energy, and we're going to take carbon dioxide. It will become reduced and fixed into these sugar molecules, for example, glucose. Also, water is going to be oxidized or really split, and oxygen gas is going to be formed. So let's take a look at some structures. In plants, photosynthesis mostly occurs in the leaves. And so if we were to take a look at this leaf, we'll find that there's this interior portion called the mesophyll. These have cells that can contain, 30 to 40 chloroplasts each. And so we also need to concern ourselves with these structures many times found on the bottom of leaves called the stomata. They're really just pores that allow oxygen to that's produced in photosynthesis to leave the cell, but more importantly, where carbon dioxide can enter in the process of photosynthesis. If we take a closer look at the main organelle involved, we'll remember that chloroplasts are double membraned organelles. They contain thylakoid discs, and if we took a bunch of thylakoid discs and stack them, we call that a granum. This is where the light reaction occurs on the thylakoids. And then the second part of photosynthesis occurs out here in the liquid portion, or the stroma. So we have the light reaction on the thylakoid, and the Calvin cycle occurs out here in the liquid portion of the stroma. If we take a look at the overview of photosynthesis, first we're going to talk about light reactions. Here is where we're obviously going to be using light. We're going to need water. A byproduct of the light reactions is oxygen gas that's going to be released. But we also will be making these energy molecules that will be important to enter the Calvin cycle. And this is where, in the Calvin cycle, in the stroma of the chloroplast, we're going to be fixing carbon dioxide into these small sugars that can then be assembled into larger sugars like glucose and sucrose. We also just keep regenerating the oxidized NADP, and that again is going to get reduced and make NADPH that's then going to enter the Calvin cycle. So this is a constant oxidation reduction reaction occurring in photosynthesis. If we're taking a look at the light reaction, let's consider light for a minute. So visible light, we consider it able to be broken up into their characteristic wavelengths we identify with colors like Roy G. Biv. And when we're over here in the red spectrum, we talk about the wavelengths being a little bit longer and also having lower energy compared to the indigo-violet region, where you have shorter wavelengths of light and higher energy. I also asked you to consider a couple of graphs. So the first is the absorption spectrum graph, and really what we're showing here is many pigments involved in photosynthesis and which wavelength they best absorb light in. And so, for example, chlorophyll A tends to absorb light best in this blue indigo region, and also down here in the red region. And then if we compare that to the action spectrum graph, that's showing us the rate of photosynthesis. So it's measuring that in the amount of oxygen that's produced. And if we compare that to, for example, our chlorophyll A spectrum, it resembles it, but not exactly, because wavelengths that chlorophyll A cannot absorb. And that's where these accessory pigments, they absorb wavelengths of light that certain molecules like chlorophyll A is unable to absorb. I also asked you to consider the Engelmann experiment, and basically this is a gentleman, a scientist from the late 1800s, well before we had the instrumentation that could easily measure the absorption spectra of certain pigments. And what he did is he took a filament of alga and he exposed it to visible light, but through a prism, so he separated the light. And then he exposed that filament to aerobic bacteria. So in other words, bacteria that clustered around oxygen sources. And Essentially, what he found is that he had the highest amount, the most bacteria in this blue indigo wavelength of light, and also down here in the red. So he proposed that the rate of photosynthesis increases in these wavelengths, the blue, indigo, and the red, which now we have confirmed with bigger tech instrumentation. As we enter the light reactions, we need to remember where we're at again. We're on the thylakoid membrane. And so here's a picture of the thylakoid membrane, and we'll separate the light reaction really up into two parts. And we call these clusters of really pigments and proteins photosystems. So the first photosystem we're going to consider is called photosystem 2, and the second one is photosystem 1. It sounds backwards, but these numbers only reflect the way in which they were discovered. The system 1 was discovered before photosystem 2. So if we take a look at a photosystem, we know that we have a light harvesting complex, and this is where our pigments are found that absorb the photons from the sun. And then we also have the reaction center complex. So this is a cluster of some special chlorophyll A molecules that are directly responsible really for the light reaction. If we took a closer look at the photosystem, we'll realize that in the light harvesting complex we have all these different pigments. So we have chlorophyll A's and chlorophyll B and carotenoids, and these are pigments that help absorb the light energy. Carotenoids tend to absorb the excessive light that would otherwise damage chlorophyll. So it's important to have those in the light harvesting complex. And if we talk about what exactly happens here, we would see that the pigments, when light shines in, the pigments get excited. So the electrons in the pigments get excited, and basically this energy flows through the pigments until finally it reaches the center, the reaction center. And this is a special set of chlorophyll molecules we call P680 because they absorb light the best at the 680 nanometer wavelength. And when these molecules get excited, they release an electron. And so in other words, we could say that P680 is going to get oxidized when it releases that electron. But one thing we know about this molecule here is that it's considered one of the strongest oxidizing biological molecules. So in other words, it is very ready to gather some electrons from another source once it kicks one of its own electrons out. And we'll find that that source is water. So you have an enzyme that will help split water and release the electrons from that splitting of water that basically is going to replace the P60's electrons. In the process, we're releasing oxygen. So this is the oxygen that's going to diffuse out of the plant that we breathe. So let's follow what happens with the electron that's released by P680. First, it's going to go to a primary electron acceptor. Then it is going to start falling down the electron transport chain. So we have a series of oxidation reduction as this electron falls down the chain. Energy captured in this fall of electrons are used to pump protons into the center of the thylakoid, into the lumen of the thylakoid. So we start to see a buildup of protons. And of course, we'll remember that we use that gradient to generate ATP, and that's exactly what happens on the thylakoid membrane as well. So as we continue on here, we'll remember that not only do we have photosystem 2, but we also have things occurring simultaneously in photosystem 1. Again, there's a light harvesting complex, so light energy is energizing these pigment molecules, which will finally reach the reaction center of photosystem 1. And here we have special chlorophyll molecules called P700. They will get excited and release an electron to a primary acceptor. In other words, P700 is going to get oxidized. But instead of water replacing the electrons, P700 is going to have electron replacement from those electrons that were falling down the electron transport chain from photosystem 2. So photosystem 2, when it gets oxidized, when that reaction center gets oxidized, it will have the replacement electrons coming from water. Whereas photosystem 1, when P700 gets oxidized, the replacement electrons are coming from photosystem 2's electron transport chain. So the electron from photosystem 1 is going to go down its own electron transport chain until finally it reaches an enzyme called NADP reductase. And here is where we're going to reduce that molecule into NADPH. That's how it happens. So photosystem 2 uses water to replace its electrons. Those electrons continue down the electron transport chain. That fall of energy is being used to pump protons into the thylakoid space, the thylakoid membrane. Photosystem 1, when that reaction center gets oxidized, the replacement electrons are coming from photosystem 2's electron transport chain. Those electrons get continued down a second electron transport chain till it reaches the enzyme NADP reductase, making NADPH. And then using this gradient of protons, they have nowhere else to go except through this ATP synthase, and just like cellular respiration, use that proton motive force to create ATP. So these are the two main molecules that are going to get used in the Calvin cycle in the stroma, and we're ready to talk about the second part of photosynthesis.